to put the two together now. So I'll do this little, I'll hit this again. You've already seen part of it. And then I'm going to go into the workshop, do that real quick. Essentially what I'm going to show you in this talk is results from my A to I editing findings and how microRNAs are related to them. And then I'll go right into how we identified our edits and how we look for microRNA complementarities. Good? Okay. I'm assuming we're good? All right. All right, so this talk was entitled, figure out what the point part is, RNA editing, and how it alters microRNA targeting. Again, I'm going to roll through this because you've seen it. You know what a microRNA is. I already talked about that. RNA editing, again. Whoa, that's not that. RNA editing is a change at the sequence level. Again, if it's A to I editing, it looks like an A to a G, right? We've already seen that. It looks literally you're turning an A into a G. It relates to cancer. Boom, seed matches, boom, that's our hypothesis. What do? We next generation sequenced RNA from 2 breast cancer cell lines, MCF7s and MDA-MB231s. Again, the MCF7s are the more docile. The MDA-MB231s are the highly invasive, highly metastatic line. All right. Now we compared the resulting mRNA transcripts to the genome to identify editing activity. And again, we found a number of transcripts where you see an A has been turned into a G, or in this case, a T into a C. Same things. It depends on what strand you're on. All right, so, wow, it's loud. Total RNA-Seq results. This was striking to me. We found around 104,000 unique edit sites in MCF7s. When we looked at MDA-MB231s, we found about 150,000 unique edit sites. And we find this is pretty commonplace. There was another group that reported fairly recently. They looked at another tissue, and they found about 1 million unique edit sites across the transcriptome. All right. So I was talking to a little earlier somebody out at the cookie/T table, right? And talking about how many edit sites there were, and I'm like, there is a ton of edit sites. And if you're looking at RNA-RNA interactions, if you're looking at RNA structural analyses, and you're not taking these into account, you are missing a very important part of the picture. All right. So we're dealing with 100,000 plus edit sites in the transcriptomes that we're looking at. All right. Now again, how did we look for microRNA complementarities? We looked for microRNA complementarities. In the workshop, I'll show this a little better. But essentially, we just looked for complementarity at the match versus, or at the edit when it's unedited, it's an A, or it's edited as a G. Some microRNAs showed a bias towards targeting editing, or their target sites being disrupted due to editing. We found about 96 of them that fall into those categories. I know I'm rolling. This is where I want to get to. I want to get to the results, or where we're at right now with this study. One microRNA in particular jumped out as miR-140. Right? Why did miR-140 jump out at us? It's highly expressed in both cancer cell lines, and it's relevant to cancer. All right. So in miR-140, when we took all of our edit sites and we looked, we find that in unedited state, they were a background. There's essentially none of those sites that could be edited could potentially serve as a target for miR-140. Whereas we find about 80 of those target sites or edit sites can serve as a target for miR-140 if edited. All right, again, miR-140 is relevant to breast cancer. Right, and the really striking thing is what we found is that in MDA-MB231s, the highly aggressive cell line, we were not seeing editing of the potential sites, whereas in MCF7 we were. Right, so what does that mean? That means in MCF7s, miR-140 can target this gene called DFF-alpha. Whereas in MB231s, miR-140 cannot due to editing. That's what we would predict. All right, so we tested that. What is DFF-alpha? DFF-alpha is called DNA fragmentation factor alpha. It's called ICAD, inhibitor of caspase activated DNA fragmentation, or DNase. DNF-alpha is bound to CAD, inhibits fragmentation. It's part of the caspase-3, caspase-8 cascade. Caspase-3 cleaves DFF-alpha from CAD, allowing it to perform DNA fragmentation. So we're looking at an apoptosis inducer. All right. And it looks like it does function 140. So what do we say? If I look at MCF7 cells and I look at MDA-MB231s and I add in anti-miR-140, right? I'm blocking miR-140 activity. What do I see? It doesn't do anything in MDA-MB231s. We wouldn't think that it would. Right? miR-140, we believe, is not capable of regulating DFF-alpha in MDA-MB231s, and we believe that's tied into cellular proliferation. Instead, when we look at MCF7s, what we see is over about 120% increase in cellular proliferation. All right? We next wanted to look at All right, so it looks like it is regulating DFF alpha. We think that it's DFF alpha is the way that it's affecting cellular proliferation. So we looked at my tried and true cell model for evaluating whether or not a microRNA can regulate a particular edit site. And I've got the system I like to use HEC293s and NB7s. What are HEC293s and NB7s? HEC293s are human embryonic kidney 293 cells. There is little to no ADAR activity in HEC293s. How do I know that? I've looked at the transcriptomes. I don't see anything. I'm not 100% sure that DFF alpha is completely knocked out, but what I find is that typically any site that can get edited is not edited in HEC293s. There's very minimal to no editing in HEC293s. In NB7s, this is neuroblastoma 7 lines, it's characterized by hyperediting. Every site that can potentially be edited is edited in NB7s. We find that characteristic. All right, so what are we looking at here? I'm throwing in my microRNA inhibitor, and I'm looking at whether or not we can affect DFF alpha levels in HEC293. Doesn't matter how much microRNA mimic I throw in, I cannot knock down DFF alpha in HEC293s. There's no editing there. When I instead look at NB7s, when I look at NB7s, and I can see a nice dose response. Right? Nice dose response of if I throw in microRNA inhibitor, can I knock down DFF alpha? Absolutely, I can. Right? So again, in this cellular context, NB7s where there is editing versus HEC293 where there is not, DFF alpha is responsive to microRNAs where it is not in HEC293s. Right? I got a clicker. All right. So what about MB231s versus MCF7? Same kind of thing. This is fairly preliminary data, but what we're seeing is that if I knock out MIR140, I can see increased DFF alpha levels in MCF7 versus I see no response in MDA and MB231s, which is what we would predict. All right. So where are we at? So right now, it looks like MIR140 can regulate DFF alpha in MCF7 through targeting a edit site, and it cannot do this in MDA and MB231s. What else does that mean? Single base substitutions caused by ADI editing can have dramatic effects on microRNA targeting. And most importantly, again, it is necessary to look at transcriptome data for accurate microRNA target prediction. All right. Again, a lot of that I talked about or alluded to earlier, but the next thing that I wanted to get into is how we're predicting our targets and how we're identifying our ADI edits. Well, that's not me at all. That's somebody else.
Yes, mine.
You know what? You want me to try to see how it goes?
Yeah, you're a better person.
I'm going to be. We'll see that you can see the name. See, mine's 2015 Transcriptomics, and his Transcriptomics 2015. So we're a little. Wow. All right, let's get started. Whoa, I don't want to reactivate antiviral software. Antiviral software is the virus. That's what I'm pretty sure of. All right. Go. All right. So now I'm going to talk a little bit about again how we identify our edits firstly, and then secondly how we identify microRNA complementarity to those edits. Never seen this slide before, ever. An A to an I to an A to a G, right? What do all of these humans? So like I've said, we found over 100,000 unique edit sites in MCF7s. Over 100, or about 150,000 in MDA and MB231. Other groups are saying there may be 1 million edit sites in a particular transcriptome, right? Those are relevant. Changes an A to a G, right? What do they do? Some of them alter splicing, right? They've been shown ADI edits can alter RNA localization, lead to nuclear retention, mRNA stabilities. They can affect translational efficiency. I'm going to say a lot of them are involved with microRNA regulation. By a lot, right? But largely, most of these ADI edits, the function's not known, but they do seem to be tissue specific, and have in very related tissues. You can have very dramatic differences, but they're reproducible differences. It's not arbitrary. It's not random. All right. So what did we do? Again, we sequenced poly-A RNAs, and then we compared our RNA transcripts to genomic sequences to identify potential edit sites. What are we doing? So we obtain our raw reads, right? Then we apply quality filters and adapter trimming. What's the trim? What's the program? I think I've got it here. Like trim fac, trim something. Then we're going to map our reads to the reference genome using bow tie. It says use top hat. I'm a bow tie guy. I can tie one. It takes me about 15 minutes, but I can do it. I've done it 3 times. very proud of my bow tie. But no, literally, we use bow tie. Top hat's a little better because it takes into account splice junctions. We've never had a problem using bow tie. The bow tie will generate some SAM, so we use SAM tools, and we remove known SNPs and a couple other steps. I'm going to walk you through. I don't know why I keep coming to push the button because I have it right here. All right, so again, we found about 104,000 edit sites in MCF7s. We found about 150,000 edit sites in MB231s. Right, so again, it's killing me. Trimco, I think. We need my graduate student here because I've never actually done it myself. Let's be very clear. But we do clean up our reads. Clearly, our quality scores go considerably better. And then we align our reads to the genome using bow tie. Top hat works too. But we get a nice interface where we get all of our reference seqs aligned to the genome. Generates us a nice SAM file, sequence alignment map, beautiful tab delineated outputs. Right? We take those SAM files, input that into, convert into a BAM file, sort the BAM, index it, and then here we identify variations. So we use generate a variation call format. So this is part of the software. It's not very complicated, but it's to look for SNPs. So we use the same software that people utilize to identify SNPs. Right, we get a variant call file, a variant call format. Right, so here the reference to the genome would have a T. What are we seeing? We're seeing a G or a C. So what we do is we generate the variant call format for all of the reads in our data set. And then we go through, and we've got an in-house algorithm that just pulls the A's to G's from the positive strand or the T's to C's from the negative strand. Right? Boom. So now we've got a list of potential A to I edits. I say this because there's not a lot of software that does this very efficiently. I think our pipelines works very, very well. We take we cross-reference that A to G set or T to C set with SNP databases. Right? Compare our variations with known SNPs. Discard any of those. What else are we going to discard? Anytime we see a A to I edit, or excuse me, A to G edit, we'll call it, or a T to C, that is 100% represented in our reads. That's not right. That's not, even though it doesn't map to a known SNP, that's fine. A to I editing is not 100%. It's never 100%. Even if you went to NB7s, the hyper editors, you never see 100% editing in your data set. So we're looking for something that's not 0% called, it's not 100% called, anything in between I'm happy with. Doesn't correspond to a SNP. Doesn't correspond to a SNP. It's not 100%, right? It's an A to G, right? Once we do that, we typically, it's not as common as 100%. We will generate files like these for inspection, right? And again, we generate a reference genome, just do a cluster W alignment with the series of reads that are outputted, T's, C's, right? Do a manual inspection, or we have an algorithm that actually checks. And so now we've identified our A to I edit set. And again, we run that same thing for MCF7. We identify 100,000 reads that meet those criteria, right? It is an A. It's reproduced. It's not a single occurrence. I should say that too. It can't be a single occurrence. Can't be all of them can't correspond to it, and it doesn't align to a SNP. When we do that, when we run that series, we find over 100,000 MCF7, about 150,000 MDA and MB231. So now we've got a large data set of potential A to I edit sites. Now what are we going to do? Now, for this purposes, we need to cross-reference that with microRNA targeting. Right? So how do we do microRNA targeting? So we take our list of 100,000 edits or 150,000 edits specific to that transcriptome. Right? We line them all up, centered on the edit site. We line them all up, and it's centered on the edit site. We take 100 base pairs, 5', 100 base pairs, 3'. We do this. We got to just generate an algorithm that'll go in mine, bring us our 100,000 back. And then what we'll do is we'll take every microRNA. You can actually, if you've done an RNA-Seq to identify the microRNAs that are specific to that transcriptome, you could just use those. We typically use all human microRNAs, and we'll run it starting at 100 base pairs back. We'll check for seq matches all the way across. So all of them align manually. We go across. I mean, we'll do it 2 times. We do it when it's unedited and when it's edited. Here's a couple of examples. So here, this is mirror 769. And again, with the central nucleotide is adenosine, which you find is what we would expect, very low background. Every so often you see a seed match in any particular position. Typically, this looks like, I don't know, 5, 10 of them would have a seed match at any particular position in that window. When that central adenosine is switched to an inosine, what we find is about, I don't know, 250. Now all of a sudden, there was never a preference for mirror 769 to hit a particular position across here. When you change that central A to an I, now all of a sudden about 250, 300 of our targets, or of our A to I edits, now have complements to the mirror 769 seed. Right? So here, there's mirror 769. Here's another one, mirror 513 is very similar. It's got even lower background. Now even more, about 300 of our target sites look like they have a complement to mirror 513 seed match. Right? So we do this. When we do this for our data set, we found about 96 microRNAs had differences in complementarity to our particular data or our targets. Doesn't matter. This is an older data set. This is when I looked at 22 microRNAs from 452. When we expand up to 2,500, we find 96. That's it. I don't know if there's questions. I would love to. This is supposed to be more of an interactive thing, but yeah. Thank you. Good.
We have 20 minutes about. Yeah.
Okay. Yeah. Okay.
How many RNA editing sites did not intersect with known mRNA? And
are you talking about which are in mRNAs?
no, how many did not intersect with known mRNAs, and did you take flanking sequence around those? And could you in fact use that as a discovery tool for novel mRNA and/or characterize new mRNA binding sites?
That's a very good question. I would say that it's a subset that actually correspond to mRNAs. We find most of our A to I editings are in transcripts from repetitive elements that don't correspond to mRNAs. And when I say most, probably 90%. So about 10% of them actually correspond to mRNAs of the actual edit sites. Yeah, I think using that as a discovery tool is a very interesting question. But again, I think that this process, it evolved out of viral and transposable element regulation. I mean, it's not arbitrary, so I don't know. But I think it's an interesting idea to use it to actually look for protein coding if you're into proteins. I don't like proteins. The bomb went off. I'm not interested. I can barely handle genomic sequence. It's very hard for me to stand here. You don't understand. I'm just kidding. But no. So the majority of A to I edits actually don't occur in mRNAs. They occur in ALU RNAs in particular are highly enriched. Line elements, a lot of line transcripts have A to I edits in them.
Any more?
Yes.
Thank you very much. It's really interesting, and I'm pretty sure this source you tested these two cell lines is coming from the invasiveness of and metastasis of breast cancer, perhaps because of the constitution we can see in these both cell lines. I'm just wondering, have you found in time yet to test all these miRNAs you shortlisted for metastatic EMT-MET?
No, so the ones and that's the problem, right? I can generate the sequence data and I can do the analyses, and now all of a sudden here's 96 microRNAs. That's the rest of your career. No, we've looked at 513, 769, and 140. 513, 769. But we've looked at that both in breast cancer and in neuroblastoma. They have a dramatic effect on neuroblastoma proliferation. Less so in breast cancer. MIR 140 seems to be more relevant to breast cancer. So we're excited about pursuing that.
But some of them are also already known, right?
Yes.
The miRNAs.
Yes. Yeah. Not for their relationship to editing. There's been very little work done on microRNA complementarity to editing.
for the nice talk.
Thank you.
I'm like, do you think this mechanism of editing of RNA, mRNA editing is kind of part of regulation, a physiological regulation in some type of cells, or to get them targets for microRNAs or things like that?
No, I think that's, so for a long time people have known that there were tons of edits. I think the first real paper was 1999, the first widespread. It's by Lebanon and Tel Aviv, right? And they found this Nature Methods paper. Hey, look, here's 12,000 edit sites, and nobody's really done anything with it. And it's again, it's RNA stability, degradation. People haven't really, well, microRNAs and RNA-RNA interactions haven't really came out. I think that's what they all do. Like I'm saying, there's 100,000 known. We don't know what any of them do. I think there's like 12 that involve protein changes, right? Change the glutamate receptor, serotonin receptor, one amino acid to another amino acid. It's all going to be RNA-RNA interaction, and I think most of it's relevant. I don't know if that answers, right? Yes.
Wonderful talk. Actually, I just wonder, you are doing the RNA-Seq in the NCF, those tumor cell line. How do you predict actually in the wild type cells how much the RNA editing actually will occur? So
It's very relevant. That's what we're doing next. We've got some patient samples, FFPE, so you can still get RNA out of that, and do transcriptomic analysis. So we're gonna take the old paraffin or paraffin embedded tissues. So we've got a ton of. So I should mention my Mitchell Center. They gave me money. Right, so we've got a great new center in Mobile, Alabama, the Mitchell Center, and they've been collecting samples forever, and it's paraffin embedded, so we can actually go into those data or into these old tumor slices, pull out small RNAs and get really great sequencing data from it. So I've got a huge panel. So that's our next step. Now that we're confirming that it is in fact regulating proliferation, we're going to start doing next generation sequencing and try to correlate the two: aggressiveness, right, stage, with miR-140 expressions or other microRNA expressions, and tie that into. Now you can't do I can't do the protein work. I can't show Western. Well, maybe. It's for now we're looking at patient samples currently, and going to look at the particular microRNAs. So I think it will. I think we'll be able to correlate aggressive invasiveness with microRNA expression.
I have one question. So when the microRNA the edited,
Yes.
so have you observed the expression never changed?
Of the microRNA itself, or the
Yeah.
we don't really see a difference in the ones that are regulating editing. Their expressions are pretty consistent. And the thing that was most dramatic to me is their expressions are pretty consistent, but you can have the edit sites go away or be present or absent in related cell lines, MCF7s versus MDA MB231s. You get it. Particular sites will be edited in MCF7s, but not in MDA MB231s. Expression's the same, but you just have different A to I editing.
Okay.
And those are related tissues, so it's very striking.
Okay, so is it possible to observe insertion deletion in the RNA edited side? Insertion deletion, I mean, observe RNA editing. This is my maybe see it.
I don't know.
No,
I'm not sure.
Okay.
So we're gonna. We're very excited about proceeding with this.
So I will follow this, Dr. Shung's question about, most of us, we're concerned about the biological function. You try to associate this one with cell proliferation. Is there definitely association there? Or is there some other biological function we can predict?
So I'll tell you what I did. So we've looked at, we've looked at cellular proliferation, we've looked at invasion, migration, chemosensitivity. Yeah. We're not seeing any differences in chemosensitivity, which I was struck. I really thought we were going to see, we were like tamoxifen, we looked at tamoxifen, we looked at a couple things. I thought for sure we would, as soon as we saw proliferation differences, we'd see a difference in chemosensitivity. We don't. We don't see any differences in migration. The two that have popped out for this analysis are invasion and cellular proliferation. That's kind of the hallmark, right? You look at You look at migration, invasion, chemosensitivity, proliferation. I mean, there might, I don't know. I think those are pretty good set of physiological characteristics. And we're seeing 2 of.
So you think this AI editing just kind of stochastic?
I think it's
or
this
really
particular
directional.
one, yeah. But I think it regulates everything. It's involved.
Okay.
It's going to be involved.
Thank you.
Okay. No problem.
So, question in 2 parts. One is, what is the etiology of the editing? Why does it happen? And second, how can it be reversed or can it be controlled?
So that's, I mean, those are very good questions. And people don't. We know ADAR's doing it for the majority. I mean, ADAR is editing so ADAR expression first and foremost. Right? If you have high levels of ADAR, like you have in MD7, everything's going to get edited. If it's overexpressed, everything's going to get edited. If you've got virtually no levels, you see no editing. But in a normal, I would say it's fairly, ADAR expression is fairly constitutive. It's in just about every tissue you have some level of ADAR for a relevant level. But the target sites change very dramatically from tissue to tissue to tissue. Right? So the profiles are very different between different tissues. Now, can you reverse that? I don't think so. I don't think it gets reversed at all. I think it's, and as soon as you say that you can't do it, then somebody will find an enzyme that comes in and wipes them off. But to my knowledge, there's nothing that once they get A to I edited, I don't think there's anything that reverts them back. Right? I think
but there must be a way, because it couldn't be just a one way that everything's going to turn into C's and G's and so forth.
Right. You change A to I. I don't think, to my knowledge, there's no way to revert. Well, I mean,
I'm very puzzled about the reverse process.
I'm sorry?
I'm very puzzled about the reverse process. There must be a loop there somewhere.
Not to my knowledge. I mean, there could be. But as far as this is where I'm at. I look at A to I's and the effects of A to I's on how that interacts with other RNAs.
We can take it off on that.
Okay.
I mean, after.
Yeah.
This is the end of this section.
Yes.
Thank you.
Morning section.